Here we go. So, good morning, everybody. Those of you who are here to learn about current research in precision medicine and addiction, you are in the right place. Feel free to come up to the front or stay where you are. My name is Michael McKernan. I'm the Director of Government and Community Relations at the Jackson Laboratory, based in Bar Harbor. Just a brief word about the Jackson Laboratory, because as I travel around Maine and speak to members of the community, I often ask, what have you heard of the Jackson Laboratory? And most people say, yeah, I've heard of Jax, and I think what they do is a good thing, but I don't actually know what it is they do or why. So I'll tell you just a little bit. So the Jackson Laboratory is an independent, nonprofit biomedical research institution. Our mission is to discover precise genomic solutions for disease and empower the global biomedical community to improve human health. And so from its base in Maine, Jax is empowering the global biomedical community through education, through its mice, and I don't want to steal too much of thunder from my colleagues, but also through its genetics research, and I'll leave that information to the experts. So in addition, Maine's biomedical research community includes institutions like the University of New England, and we're delighted that Dr. King is with us today, as well as Maine Health Institutes for Research, the University of Maine, and many others. Several of these institutions have projects focused on the biological origins of substance use disorder. And while broad understanding of the biology of substance use disorder is critical, research is increasingly focused on personal genetics. So the last Voice of Recovery reflected on her own personal story and how perhaps her personal biology had an impact on how she experienced the disease. Including genetic background, including environment, these things come together to provide a more holistic picture of the complex biology of substance use disorder. So today we have assembled an expert panel to share some of the important work they are doing in their own labs and centers. As a panel, we know that this is a very heterogeneous audience, including fellow researchers, some physicians perhaps, and others who have some knowledge of these topics, but then there's probably some others in the room. We're very happy to get out of high school biology with a passing grade. So we're going to try to be aware of that and present information in a way that's interesting, engaging, and encourages you to ask questions. Everybody here on this panel is a teacher by heart, so please don't be shy. Ask any question that comes to mind. So to make that easier, what we've done is we've have a Slido opportunity, which means that using your device, you can log on to Slido using that hashtag and then type in your question anonymously. If at the end you wish to come up and use one of the microphones and ask a question in person, that's perfectly fine too, and I will moderate the questions from the podium. Okay, that's enough from me. I'd like to just run through who's on our panel today. First speaker is Tamara, Professor, Department of Biomedical Sciences in the College of Osteopathic Medicine at the University of New England. Our second speaker is Jason Booyer, Senior Research Scientist, Center for Addiction Biology at the Jackson Laboratory. And our final speaker is Vivek Kumar. Vivek is Associate Professor, also in the Center for Addiction Biology at Jax. So each speaker will present, and we'll open the floor to questions at the end, and we'll get going. Dr. King. So, at your advance. Yeah.
All right, thank you very much. Those chairs are very, very tall. It is my pleasure and my privilege to be here and be a part of this panel. I'm looking forward to a discussion and hearing people's questions. As mentioned, I'm a professor at the University of New England, and I'm the director of the Behavior Research Core. So today we are going to be covering some research learning objectives to really start to talk about the biology underlying substance use disorder. So where do I come in? My research is actually on pain and chronic pain. And as many people are aware, those pain and substance use disorder often intersect. And so I want to talk a little bit about the biology, what we know, and maybe what we need to learn about that. So an important aspect of substance use disorder is that substance use can be driven by changes in pain, in isolation, and negative affect, or negative emotions. But what a lot of people don't often know is that substance use by itself can actually induce pain. So opioids can create something called opioid-induced hyperalgesia, which is a hypersensitivity to pain, and this develops during use, so while the opioids are being taken, and if you withdraw the opioids, that pain and any pain getting treated becomes worse. Alcohol use is also associated with development of pain, and again, when you remove the alcohol, the pain gets worse. And more research is finding that cannabis use also can induce a state of pain, and again, withdrawal is associated with heightened pain as well as emotional pain. And so what happens is you try and quit, you remove this substance, or you stop taking it because your pain is supposed to have gone away, and you're in pain. So what do? You go and take the substance again. And of course, this creates the negative spiral that so many people talk about. So as I said, my research focuses on pain. So why? The burden of chronic pain is enormous, okay? So according to a recent analysis, about 1 in 5 United States adults have experienced or reported chronic pain. That's an enormous number of people, and it's larger than heart disease, cancer, and diabetes combined. The financial burden is enormous as well. Over $600 billion annual is spent on dealing with pain and chronic pain. This is medical costs, but also loss of productivity. And again, this is exceeding the costs of heart disease, cancer, and diabetes. So it is a pretty major problem. And in order to deal with this problem, what we need to do is learn more about it, because we just don't have a full understanding to adequately prevent chronic pain or to treat chronic pain. Treatment of pain is currently done pharmaceutically through 2 broad classes. Many people are familiar with the first one, opioids, right? So this would be your oxycodone, morphine, fentanyl. However, one emerging problem is that the rise in opiate addiction, overdose, and deaths have led to a significant reduction in appropriate opioid prescribing, leaving many patients in pain because there are no good alternative drugs. What are the alternative drugs? Those are the quote-unquote non-opioids. These span a lot of different classes of pharmaceuticals. So most people are familiar with NSAIDs like ibuprofen. Gabapentinoids and SNRIs are used to treat chronic neuropathic pain and is effective in some but not all patients. And some people can use peripheral nerve blocks such as lidocaine or capsaicin creams or patches. However, these are not optimal, and there are many patients, even on these therapeutics, that remain in pain and are seeking to alleviate that pain. And so what are we doing? We're looking for the answers. To do that, we need to build and expand upon what we already know. And we actually know quite a bit about the neurobiology of pain. So we have discovered a lot about the molecules that transduce the environmental signals to a neural signal that transmits this information through the spinal cord to the brain. We've learned a lot about how this signal is processed, leading to the perception of pain, how it's modulated, how the brain can amplify or reduce pain depending on the context or the situation. If you're running away from a bear and you scrape your knee, you don't want to be focusing on the pain. You want to be focusing on getting away from the bear. We've learned a lot about the endogenous mechanisms by which that can happen. One thing that's becoming more and more evident is that opioids and cannabinoids overlap in a lot of these areas and a lot of the biology underlying the neurobiology of pain. And so they are found in the same peripheral tissue, the spinal cord, the brainstem, the limbic structures that mediate the emotional component of pain, and cortical structures that are important in decision making. Do I escape? Do I avoid? What do I do? And what do I do in the future? They are also found in the reward pathways. So one thing about pain is very, very complicated, right? And we've learned a lot about the sensory discriminative aspects of pain. These are the things like the intensity, the duration, location. But we still need to learn a lot more about the affective motivational aspect of pain. This is the unpleasantness associated with pain. And this is what motivates us to escape, avoid, and teaches us, don't do that again. Pain relief feels good. So pain relief has been shown to elicit reports of that feels good in patients. Moreover, it activates the reward pathways in the brain of both humans and animals. And so we can use this information to better assess and create better tools to figure out what's going on. So the motivational aspects of both pain and reward are included within the same areas of the brain. And so this creates complications when you're talking about the intersection of pain and substance use disorder, because they impact one another and your brain changes in response to both of these inputs. Both humans and animals will escape something that they find painful, and both humans and animals will approach something that provides pain relief. If I have a headache and I'm a migraineur, I have learned over time what medications help. Caffeine helps, blocking bright lights helps. Okay, and one of the things that I've done in my research is I've applied these concepts to animals to show that pain relief in animals, in our rodent models of chronic pain, can create a preference for a novel context that has been paired with pain relief. And this happens both in postoperative models, but in chronic pain models too. And this opens up a whole new arena in which we can learn more about what we don't know a lot about, the emotional component of pain. We've also shown that pain relief will actually actively activate the reward pathways. And this can allow us to gain more interactions of how does this change in the context of chronic pain? How does this change in the context of chronic substance use? Maybe we can get a handle on this and treat both. And so this is the goal. If we can improve our treatments of chronic pain or pain in general, maybe we can come up with alternatives that are more specific, more comprehensive, and doesn't leave people in need and in search of alternatives. We also need resources to do this. And the NIH has put forth something called the HEAL Initiative. And this initiative has two primary goals. One goal is to improve understanding, management, and treatment of pain, because this might serve as a powerful prevention tool. The second goal is to improve the prevention and treatment of opioid misuse, addiction, and overdose. And this is happening at a very lucrative time. We are really in a new era of research, very exciting times. So we have advances in genomics and proteomics that is giving us so much data on molecular entities and pathways that may be optimal targets for both pain and substance use disorder, and maybe allow us to figure out where do these intersect. Many people are turning to pharmacogenetics to try and figure out how do we create more individualized and specific therapeutic options for patients. Because as you heard earlier, pain is an individual thing, substance use disorder is an individual thing. And there are biopsychosocial models that try and take into account these different aspects. I work on the biology, but we can't ignore these other aspects because they impact, guess what, the biology. Finally, the emergence of artificial intelligence is getting better and stronger, and this is a critical tool in analyzing the enormous amount of data that are coming in, both with the proteomics and genomics, as well as looking at, I think it's the next slide, using large language models to determine and analyze what patients are saying about their pain, so that we can get a better understanding of what they're going through. And that might lead us to novel targets within the brain that we can better understand this. And we are also using these tools within the preclinical setting to better analyze behaviors that we might associate with pain. And this is true in humans as well as in animal research models. This is going to allow a higher rate of translation from the bench to the bedside, and hopefully get tools in people's hands that are trying to treat both of these issues. And so with that, I will end and allow my colleagues to speak. I'd like to acknowledge the federal funding that I received from NIH. I am part of the NIDEA Award, which is to expand research opportunities and infrastructure to states that don't receive as much funding as our close neighbors. And we have built a center of excellence for pain research that I'm a proud member of, and we hope to keep growing. Thank you.
Good afternoon. Thank you for joining us here today. I'm really excited to be able to address you and talk to you about some of the research that I'm doing at the Jackson Laboratory. But first, because this is Maine and we're all 2 degrees away from one another, let's see how we might be even related. I went to the University of Maine at Farmington, so go Beavers. Nice. And a shameless plug for my nephew, who's a nature photographer right there. But I went to do my graduate work at Marquette University, where I got my PhD. Go Golden Eagles, not so exciting. But my family is many generations from western Maine mountains up in Phillips near Farmington. And it's my pleasure to have a research career in academia. I come from a working class family with very blue collar roots, first generation college student, never mind PhD. So I'm really just an honor to be here. And now I'm showing you a tantalizing view of where I spend my time, and this is the Jackson Lab, a nice view of it. A lot of people don't know it. It's kind of nestled there amongst a national park. It's a great recruiting tool to get people to come live with us. We don't show the snowy picture, but here's an example. So as Dr. King mentioned, opioids can do a lot of things, and some of the things that they do are analgesia or pain relief. But they also target respiratory depression, cardiovascular depression, decreased gastrointestinal mobility or constipation, sedation. They cause euphoria, which is why you keep going back to them, and then urinary retention. Now, all of these things have common mechanisms that are controlling them, such as the opioid receptor. That would be an obvious thing that opioids would bind to. But each of these traits is biologically distinct, and it is possible to actually identify pathways or molecules that will affect analgesia only, but not respiratory depression, or will affect euphoria, no euphoria, and just give you pain relief. So the idea would be to separate these different effects of opioids. Now, as I mentioned, the biological basis for overdose. So I said respiratory depression, that's the big thing that I study because that's basically what people are dying of and you're using naloxone to prevent. You get a bunch of cells in your brainstem. They recognize the fact that you're not breathing well and you're getting low carbon dioxide and the high carbon dioxide and low oxygen, and the opioid causes them to not increase your respirations. Instead, they go down and down and down. And as a result, you're going to have opioid-induced respiratory depression and death. Now, reversal. As we've mentioned, naloxone, this is a great molecule, and I'm so glad we're getting it everywhere. And here's just showing you a little picture of a schematic where the opioid binds to the opioid receptor, but naloxone also binds to the opioid receptor and kind of displaces it. And we designed it so it actually binds tighter to the opioid receptor than classical things like heroin or morphine. But a caveat here is, and that's something we need to be trying to stay ahead of, and as researchers we're always trying to stay one step ahead, is that these newer drugs such as fentanyl, carfentanil, they bind also very tightly to the receptor, so you may need even more naloxone to get the effect. So we need to look for reversal agents to supplement this, in addition to continuing to get this widely available. All right, so at the Jackson Laboratory we study mice. Why do we study mice? Well, mice are genetically related to us, similar, about 92%. But the human population, we're all related to each other about 99.999%. And I'm representing the diverse population here by these minifigs. Now, each set of minifigs carries with it their own set of genes, and so we also have a population of mice on the left that is like a population of humans. So each one of those is supposed to represent a mouse minifig or a mouse individual. So now we have mice that are also 99.9% alike, and we can look at them and say, okay, what are their genes in their genome that's causing them to be extremely sensitive? So that bottom line shows that one half of a dose is all that's needed to cause overdose. The middle line shows a full dose is needed for overdose, and then the top shows a lot is needed for overdose. And so this is a very important diagram because we don't know what we're walking around with for alleles, and you don't know what your person you're using with has either. And so you're sharing a dose with them and use the same thing that they used. I can see where it could lead to problems. Within the mouse room where I can control the dosage and I have very pure compounds, there's a 150-fold difference in sensitivity between one mouse and another mouse, only looking at 8 different mice. Now imagine if I look at more mice, how diverge, and that's for fentanyl. Morphine, it's about a 4-fold difference, and morphine and fentanyl work differently even though they're both opioids. All right, so using this system, I was able to identify loci or regions of the genome that when you vary them, you get respiratory depression or you don't. And that was using mice. And then humans have taken and looked at clinical data for using a trait is basically how many times somebody has overdosed and been brought back is the trait, and see how likely, use that as a risk score for how likely they are to overdose. And in those genome-wide association studies, they've been able to identify regions of the human genome that also vary with this particular phenotype or this particular symptom, overdose. So here we have humans and mice, and we can look for overlap between the two of them. Now, why do we care about this genomic knowledge? Dr. King got into some of the pharmacogenetics, pharmacogenomics uses. Here, this figure lists, that's stolen from a website there, lists a bunch of different uses for the allele, the genetic knowledge. The example that I like to give is being able to know if you should or should not be prescribed an opioid based on your risk. Like, this person is so sensitive, we shouldn't give them that. They should have something else. Or if somebody is using how risky is this person who's in your clinic in front of you, can you give them a warning, a heads up, like you're kind of a sensitive individual, and they may already know that, but it's just knowledge is power. Emerging threats, as I said, we have to stay one step ahead. So right now on the street there's xylazine and fentanyl mixed together called tranq. This causes nasty wounds. And xylazine is something I was always familiar with since I've been at the Jackson Laboratory because it's a veterinary anesthetic. We use it to put down mice to do surgeries. And as a result, there is no known reversal agent for humans because we don't use it in humans. And so there's nothing approved to reverse. It binds to a beta-2 or alpha-2 adenoreceptor, not the opioid receptor. So naloxone is going to help you with the fentanyl threat, but not the xylazine threat. So there's where we need to look for another intervention or another angle. And again, something else that's also coming out are these nitazines and derivatives. And by derivatives, I mean they'll just add a methyl, ethyl, or a little, an extra carbon here or extra carbon there, and make different variations of the same drug with extremely different potencies, ranging from 1 to 100 times that of fentanyl. So these are the emerging threats, and we can use this approach to try to find out which genes are involved in those pathways, and those could become targets for either screening or interventions, like what is the human body, what is nature showing us is a mechanism to make you more tolerant to a drug? How can we make somebody else more tolerant to that? So we're kind of using the natural variation in the population to inform us about the biology underlying these traits. So the key takeaways, and something that I was glad to hear the doctor at Millinocket Hospital indicate, that addiction is a disease, and it has a biological basis, and it's not a moral failing. I have mice that love to use, and I have mice that don't want to touch this stuff. What did they do? They didn't go to church on Sunday? No. There's a basis. It's often we talked about these comorbidities. Some of these mice also do have signs of depression, if you will. So there are comorbidities that associate with addiction susceptibility. But like addiction, opioid overdose has a biological mechanism, and if we can understand the mechanism that causes the difference, we might be able to dig in there and find a new target or something to help us help with the situation. And then finally, monoclonal organisms give us the ability to try and separate out the different functions of the opioids. For example, separate that pain relief away from the opioid-induced respiratory depression, or as I like to say, stay high but not die. That's my bumper sticker. All right, that's it. Thank you.
Good afternoon, everyone. My name is Vivek Kumar, and I'm going to build on what Dr. Boubier and Dr. King talked about and give you some concrete examples of how genetics shapes responses to drugs of abuse. So we should think about addiction as a cycle. Essentially, individuals start off by consuming drug on a social basis, drinking on the weekends, having fun on the weekends. And then slowly it kind of cuts into the weekday. That's kind of this escalation, binge drinking in terms of alcohol. Then the person becomes dependent. When they don't consume the drug, they don't feel well. And they may realize this, that I may be consuming drugs too much, and they may want to stop. And when they do that, their body says no, and they go into withdrawal. That is a very unpleasant feeling, and person will likely relapse. They'll start using again. And this cycle then continues. A few individuals will go into protracted withdrawal and then recovery. And they, hopefully will stay in recovery to kind of break that cycle. And on the left, we talk about kind of what affects this cycle. So genetic variables, we saw good examples here, affect that cycle. Environmental factors affect that cycle, such as stress. And a lot of conditioning effects affect that cycle. So for instance, certain places are in our brains are associated with drug consumption. And a part of recovery is kind of having some sort of behavioral cognitive therapy to kind of break those habits, right? And so I always use the story. I had a colleague who posted on Facebook that, she hadn't smoked cigarettes for like 20 years, and she put on a jacket. She's had a leather jacket for a really long time, and the first instinct was to reach for her cigarettes in her pocket, right? Same thing, if someone is using drugs in a basement, that place is conditioned with the drug response, and as soon as they kind of go into that basement, there's kind of this, the brain activates its memories, and they're like, oh, I should use drugs. So a lot of conditioning effects are also responsible. So why do we use mice as a model organism? So Dr. Boubier covered many of the reasons. Genetics is a really important one. I see the slides are slightly, okay. Our brain circuits are conserved. Obviously, mouse brains are smaller, but we have essentially the same cell types, the same connectivity between brain regions. And we can, if you were to see it, you would see we can control their environment. We can control we can essentially do experiments in these animals at great detail. Okay, so one of the assays that we use is sort of a locomotor activation assay. So let's see, maybe not. Well, yeah, okay. The mouse on the left is not moving, but essentially the mouse on the right has been administered a cocaine, and it's a stimulant. So mice, get hyperactive, and we can use this as a sensitivity measure to the drug. We can, of course, do much more complex experiments, like teach them to press a lever to get the drug, and do more kind of self-administration type paradigm. So with that in mind, I want to give you an example of how a single nucleotide, one base pair in the DNA of the mouse out of 2.8 billion base pairs, can actually affect sensitivity and addiction vulnerability. And these are things we can actually do in mice, but are very challenging in humans. So in 1951, a colony of mice from Bar Harbor, Maine, were shipped to the NIH in Maryland. And these were essentially identical mice. So imagine if we had 100 twins, not twins anymore, but clones, and we shipped 50 of them to one location, kept the other 50 in the original location. And these mice are just, they're breeding amongst one another, but they're essentially identical in 1951. But because the process of DNA replication has errors, it's a process known as genetic drift, there are kind of subtle changes that are picked up. So we then got mice from Bar Harbor and from NIH, and we tested them in 2013 for that sensitivity to locomotor response, right? So nothing has really changed in these. The mice have just kind of picked up a few changes through this kind of process of genetic drift. And it turns out that response is just very different. So I've just condensed a lot of data in about 2 years worth of work here. But the Maine mice tend to have that high locomotor response, and the Maryland mice, the NIH mice, tend to have that low locomotor response. But so then as a geneticist, I can ask the question, well, they were identical in 1951. What has actually changed in 2013? And this is a process known as genetic mapping. We do a lot of crosses, we do a lot of sequencing. And from that, we're able to identify one nucleotide change, and that's that in the Maine mice, you see that TCT, and that codes for phenylalanine, and that changes to TTT in the Maryland mice, and that codes for serine. And that happens in a very specific protein called CYFIP2. It's a complicated name, you don't need to know it. And that is actually regulating that difference in cocaine response, right? So what we've essentially done is identified a pathway and a novel allele or a novel mutation that regulates this kind of cocaine response. Now we can actually ask a lot of interesting questions. So we then we used, again, the resources of JAX to create a precise mouse model. So we went in and we took mice that didn't have the mutation and we created that mutation. And we took mice that had the mutation and we corrected that mutation. So now we have kind of 4 strains or kind of a reciprocal change. And we then shipped it off to colleagues who are really good at studying nicotine. In University of Maine, Alan Rosenwasser and his students study alcohol drinking. And then at Boston University, I have a colleague who studies binge eating. Now, it turns out that gene actually regulates all 3 of these phenotypes, right? So going back to this idea of overlapping circuits that Dr. King brought up, the brain, there is no one circuit for taking cocaine or nicotine. It's a common circuit for reward. It's used as a survival circuit, and it regulates many things. A reward to drugs of abuse like nicotine, cocaine, alcohol, but also reward to natural rewards. Food is a natural reward. Sex is a natural reward. Exploration, exercise, these are all natural rewards. And in therapy, we try and substitute a non-healthy reward with a healthy reward. So these are the kind of advantages we have with mice, and these are the kind of experiments we can do with mice. In fact, so we discovered this back in 2013. Over the past 3-4 years, there have been a slew of human genetics patients with mutations in the same gene called CYFIP2. Not the same mutation that we found in mice, but in a different region in that same protein. And it turns out that these are individuals with intellectual disability, epilepsy, really severe kind of developmental disorders, and in many cases they die. They don't make it past some development, and these are human mutations. So what does that tell you? So really that gene and that pathway is serving multiple functions. If you have a certain mutation, you have a reward dysfunction. If you have other mutations, you can have very severe neurodevelopmental disorders. So again, advantage of using mice to dissect these complex traits. So so this is just meant to show, so this is the brain, and the different parts of the addiction cycle are regulated by different parts of the brain. But it's a very complex system, basal ganglia or mesolimbic reward circuit, and we really need to study, and we need to kind of, now that we have a gene and a pathway, we can go in and perturb the gene in specific circuits in the brain, and we can determine its function in each one of these parts. So the next part is, Dr. Boubier talked a little bit about this, and Dr. King too, in terms of, can we predict who's at risk, right? This idea of prediction, prediction. So this is an old field. So, we know that there are traits that teenagers show, such as impulsivity, anxiety, sensation seeking, and hopelessness, and these put kids at risk for addiction. There's newer kind of predictive algorithms out there. There's a machine learning one that on the right that was covered by the New York Times that pretty much, it's a proprietary system, so we don't quite know what's in it, but the idea is when you go to your doctor, the doctor uses this machine learning app and predicts your addiction liability, and if you meet a certain score, you're not going to get prescribed opioids. But there's a lot of even polygenic risk scores based on our genetics. Can we predict what the person is doing? So again, here's where it's a very non-precise field, I would say right now. I would not use these black box algorithms, or even polygenic risk scores are not very accurate right now. But in mice, we can again work on a lot of these. So we can observe behaviors in mice. Can we, based on the behavior and the genetics of the animal, predict which animal will likely show uncontrolled drug intake behavior? So mice, we can observe them over long periods of time and say, well, if the mouse is fighting, or if the mouse has certain behavior, is that mouse more likely to consume drug? So for this, we've kind of again turned to machine learning or AI methods. At JAX, my lab has pioneered the use of kind of digital phenotyping, and we built these very fancy home cages where there are cameras and mice can live in it, and we essentially are, like a big brother type observing these mice for weeks and maybe entirety of its life. So this is what kind of a view of a camera looks like. Here are 3 mice kind of happily living. There's a wheel, some nestlets and things like that, and we can then use this to kind of extract behavior from this. And this is a process that uses machine learning. We don't need to go into this too much, but essentially we take the raw videos on the left and we end up with kind of abstracted images of the animal. Then we can go from that to look at animal behavior. And so I feel like this, and we can then correlate that behavior with drug intake behavior, with genetics, or many other things. So this is, I think, a new field that, in my opinion, is going to have a lot of impact. Okay, I'll stop there. I'll just say one thing, which is funding. For my postdoctoral work I've been funded by NIH. It was great to see Shamsha here. I think, the National Institute of Drug Abuse and the funding that they brought into this area is really important and deserves our support. Thank you.
Okay. Hello. All right. Thank you all. Fantastic introduction to the topic of the day. We have time for some questions, and I've asked that so the slide is back up there. Thank you. And feel free to use your device to go onto Slido, or help yourself up to one of these 2 microphones, and we can start our discussion. So, oh boy, I think I lost my my slide here. Anyway, I showed you the question earlier, so we'll remember it, right?
Of course.
Okay, so somebody asked a question regarding cannabis, and I have to log in to find that question again because it logged out. Do you recall?
I think I remember, yes. Hi, so it was an excellent question, and I actually have a little story about the question. So the question was that I had mentioned that cannabis can be addictive and lead to substance use disorder, but that they had heard that cannabis is not addicting. And this is very, very common. In fact, I worked with my daughter, who just graduated high school this past year, on educating their peers on this very topic because so many were coming up. Oh, well, here's TikTok, and TikTok told me cannabis is not addicting. Cannabis is legal. So we know everything about it, right? We know how safe it is and everything. That was my sarcasm hat. And so, I think this is very common, and I don't think it's an easy yes or no answer. We just heard a tremendous amount on genetic variability, individual variability, and I'll remind you that biology is one component of a complex system. So we have genetics and biology, we have social determinants, right? Social networks, what's going on in that person's life, what's going on at school, and we have psychological factors all contributing to whether or not a substance could be addictive. And so I'll leave it with that. I'm sure my colleagues could also answer.
Excellent, thank you. So I'm back online. I can now see your questions again, which is very helpful. So a question came in, is there any research into the impact of mental health symptoms and the perception of pain?
Yes. So that is ongoing and a fascinating area. And as I said, we are kind of moving into gaining a better understanding of the emotional, motivational, cognitive aspects of pain. But there has been a lot of research on things like effectiveness of cognitive behavioral therapy for people with chronic pain. Has been shown to be very effective. Combination therapy, exercise, CBT, and that kind of thing has been found to be helpful with patients with chronic pain. And there are also patients who tend to be more susceptible to chronic pain and more resistant to therapies, especially there's something called catastrophizing, which can complicate treatment of these patients quite a bit. So yes, that was the long answer for there is definitely a lot of ongoing active and forward thinking on the impact of mental health on chronic pain and the impact of chronic pain on mental health. Depression, anxiety are comorbidities that are very common.
All right, I'm going to go down. Vivek. Oh, I'm sorry. Thank you. Go ahead, sir.
Hi, this is for Dr. King. First of all, I'm Paul Vinsel. I'm a alumnus of St. Francis College and Unicom. So,
Very nice.
Go Nor'easters. You talked about opioid-induced hyperalgesia, and I'm curious if buprenorphine can also produce opioid-induced hyperalgesia.
Great question. So buprenorphine, it's a polydrug, and so I think basically what it's been shown is that it's less likely to induce the opioid-induced hyperalgesia. Whether it will actively block it or not, I don't remember off the top of my head.
All right, lots of questions are coming in, so thank you. I'm going to go now to the other end to Vivek, since you were attempting to show a video of cocaine-induced behavior. So if we were to perturb the gene that regulates cocaine response, could this lead to research or avenues for medication-assisted treatments for stimulants like cocaine and nicotine?
Yeah, I mean, that's exactly the idea, like to develop and find novel genes and pathways that can be targeted for therapeutics. In the case of CYFIP2 that I showed you, we have to think real carefully about it because overactivation of this pathway can lead to kind of that neurodevelopmental disorder or seizures. Underactivation or kind of loss of this pathway, at least embryonically, can actually be lethal. Mice that have two mutated copies of this gene or deleted copies will actually die. So it's somewhere in the middle that we have to titrate, for this pathway. And that's kind of fairly common. I mean, as I mentioned, there is no specific gene for cocaine use. So we have to think about, like how these drugs may affect other phenotypes.
Great, thank you. Go ahead.
Yeah, my name is Lynn. How many decades ago did they actually, within the mice and with humans too, did they narrow down a specific gene that's addictive receptive, So why one person is not an addict and another is? Had anybody narrowed down the gene? Because we all know it starts out with genetics.
So there is no the gene. It's it's hundreds of thousands maybe of genes are involved, each with small amounts of changes, and that's why it's a very tough thing to determine.
Yeah, I just want to add to that it's kind of like many roads, one destination, and I recently did a study with like 33 different mutants, so 33 different genes we knocked out, and we had screened them in advance and predicted that some of them would be associated with addiction or compulsion. 17 of them were, and then we looked at the pathways, and none of those 17 pathways overlapped. So that was 17 unique roads to one destination.
So this is another question for the mousers. Somebody in the audience was curious about the environment in a cage, in a container, and what else you would find in a container in terms of a behavioral assay for mice. Are they alone? Are they with groups? I've heard of enrichment. Can you talk about that a little bit?
They're all of the above. It's really up to us. So we have mice in shoebox cages where we're just using them to breed. There's not a lot of space for them to do enriched behavior. We have mice in 2-foot by 2-foot arenas with mazes and toys where we allow them to hang out with their friends and do all the actual cognitive tasks and things like that, or administer drugs. So really, that's the nice thing about using these this amazing organism is we can customize the environment and the animal to the questions we're asking. You want to talk about your Lego, the operant Lego box you guys built? I think that's a really cool place of.
Oh yeah, so one of this classic in vitro self-administration assay, or are mice giving themselves drugs, is in an operant box or a Skinner box where there's 2 levers. And using the Lego Mindstorm robotics kit, a high school student came into the lab and built out of Legos a Skinner box with 2 levers that dispenses a food pellet when they press a certain lever, and then you could program the computer so that they have to push the lever 3 times, work harder, or they switch the levers up in the middle. They figure out it's the other lever. So we could do all those experiments using Legos, and that was much cheaper than scientific equipment.
So, a couple questions here about psilocybin, and they're distinct, but the one that I want to ask is the following. Is the idea that the brain doesn't really know the difference between when somebody uses opioids versus psilocybin or marijuana accurate? Can the brain distinguish between those 3 very different substances? Whoever wants it.
I'll take a crack. So there are billions of neurons and millions of pathways, and they can really finely distinguish between experiences and learning. So they all have different effects. So I think if you were to take somebody, instead of Coke and Pepsi, do these different drugs and a taste test, they would be able to tell you that they have different experiences. They might not be able to tell you which drug they took. And I will do one last thing. For example, cannabis, 151 different strains. What you get in whatever you're taking or administering depends on what strain and where it's harvested and how it's processed. So even within cannabis, you could have 3 different things of cannabis and have very different responses.
I was just going to add that, like, cannabis has cannabinoids and they bind to cannabinoid receptors. So nicotine binds to nicotine receptors. So there's this specialization within, and it's not just one receptor, there's a family of these receptors. So those levels can be higher or lower in some cells and not in other cells. So it really is a moving target that we're after, but we're trying to find ways to find it.
All right, we have just time for maybe 1 or 2 questions, depending on how long the answer is. So xylozine's been mentioned a couple times. The question is, where is the research with creating a naloxone-type treatment for xylazine? Is there a projected date for which they hope to have a response treatment available to the public?
I'll just say that prevention, of course, is the best measure right now, and we have the test strips for xylazine, and Maine was one of the first states, I believe, to actually push for that. And so they are available. The National Institute of Drug Abuse, which Vivek mentioned, is very interested. So I speak to my program director a lot, and he's like, okay, where's your protocol for your proposal for xylazine? You got to realize that science is kind of glacial and it's moving with regulations. It can take like 18 months to go from an idea to money. So that's kind of where it's at. I have lots of ideas.
Come see him afterwards. Okay, final question, and there are a couple questions here about children. So does age, parentheses, brain development, have any impact on whether or not someone becomes addicted to substances? For example, is an adolescent or young adult more susceptible to SUD?
Yes, I talk about this quite a bit. The same drug in a 10, 12, 13-year-old it will have a very different effect than that drug on a 23, 25, 40-year-old, right? So, I think of it as like a rainstorm when your house is incomplete and it's exposed and it's being constructed. It can do a lot more damage than when the house is fully constructed. So yes, exposure, and when does the brain stop developing? In men, it's around 25, 26 years old. So the drinking age should not be 18, 21. It's 18 in Europe and 21. It should really be 25, 26. I mean, insurance companies figured this out, right? This is why insurance rates drop when you hit 25, because men don't make stupid decisions as much anymore. So yes, the answer is a big yes. I think it's really important that the brain, the developing brain, not be exposed to even things that are quote unquote thought of as non-addictive, like cannabinoids or hallucinogens or things like that, because they will alter circuits and have long-lasting effects.
The questions in science are endless. Thank you so much for your attention. Please come back afterwards and chat some more. Thank you to the panel.